We're in the digestive system. In this lab, we're going to study the digestive systems, the organs in the digestive system. Your GI tract digestive system is a long tube. It's about 9 meters long. So in this tube, you have a lot of structures. We're going to look at those structures. And we're going to also look at some microscope slides, histology slides. Your digestive system starts from your mouth and to the esophagus. So inside your mouth, you have the salivary glands produce saliva. So this is a picture I copied from your textbook. So these show you all these structures in your mouth, mainly for mechanical digestion. And those chemical digestion come from the saliva. So you have the saliva gland produce saliva. And the bolus, after the food mixed with the saliva, and it becomes bolus, will go through your esophagus and will go to the stomach. So the stomach have these structures. So these are the stomach structures. And the rogae are the folding structure inside the stomach. So the stomach inside is not smooth, you have those foldings. And the pylorus is the sphincter, so it will hold tight for the food to stay there. And after digestion finish, it digests the protein, and this will open. Now the food go to the, we don't call the food, we call the chyme, chyme, chyme. It will go to the small intestines. And the small intestines is about 7 meters long tube, can be divided into the duodenum, jejunum, and ileum. So it's 3 different parts. Duodenum is about 12 fingers or 12 centimeters, 12 fingers put together. That's the distance. Very short, and that's the last place for digestion to happen. And the duodenum and jejunum and ileum, these 2 places are for absorption. And all this long tube in your abdominal pelvic cavity, they were fixed there by the mesentery. So mesentery are those peritoneum, and they're like duct tape, but they're much more than duct tape. They fix the GI tract in the correct position. And these are the large intestines. So you use your textbook, identify this structure. These are pretty self-explanatory. And yeah, we're gonna go look at the liver and pancreas and look at those ducts. Okay, now let's use this to explain. So your liver is around here. Your liver produces bile, and the biles will be stored in gallbladder. So gallbladder is a container. After the liver produces bile, they will go through the right hepatic duct and left hepatic bile duct. Will go through these 2 and go to the gallbladder, and they wait there. Gallbladder is a container. And when your chyme go from stomach to the duodenum, go to here, and it will trigger the reflex, and it will make the gallbladder produce bile. So you go to the cystic duct and come out to here. When you come to here, you found you have another duct called the pancreatic duct. These 2 actually have share the same opening. And the pancreatic duct gonna release pancreatic juice. So together, the bile and pancreatic juice together will go through the duodenum and for digestion function. And the last one we're gonna look at is those villus model. So in your small intestines, you have a lot of finger-like structure called the villi. And the main purpose is to increase the surface area. So all your GI tract have 4 layers: mucosa, submucosa, muscularis, externa, and serosa. So the inner layer is mucosa. Mucosa is the inner layer. It touches the food directly. And the inner layer is not smooth. They have a lot of folding, like finger-like structure. We call them villi. And their function is to increase the surface area. And these villi in your small intestines, they do a very good job to increase the surface area. So when we calculate the total surface area of your small intestines, is about a full tennis court is close to 2,400 square feet because of those villi. So it's in the mucosa layer. And the second layer is called the submucosa. And the muscularis, that's the third layer. So they always have two layers, smooth muscle, because one need to mix the food with digestive enzyme, and the second one is actually push the food from the top to the bottom. So they have two layers, circular layer and longitudinal layer. So these are the four layers. And this picture show you, okay, in your small intestines, this is your mucosa layer, this submucosa layer. And in the mucosa layer, you have those villi. And let's look at the liver. Liver has more than 200 functions. And one function in the digestive system is it produces bile. It also helps to clean up the toxic molecule from your food. So the food after absorption will go through the hepatic portal vein, go to the liver. So liver is supposed to clean those toxic molecules. And liver has a lot of functional units. There are millions of them called the lobules. So lobules are those hexagon structure. These are the lobules. So in your liver, you just have a lot of them. Each of them serve exactly the same function of the liver. So they will clean the toxic molecule and produce bile. So if I give you this liver slice and ask you what's this, and the answer will be pretty obvious. That's the lobule structure. Okay, that's it.